Okay, yeah. So everyone, welcome to the seminar here today. It's very honored to have our speaker, David Valen from Caltech. So David is, I first know David's work is several years ago because my lab working on lifestyle imaging, we need to segmentation, we try to use the machine learning, right? CNN, those methods. Then we noticed David's paper. So that's where I started to notice him. Then later I know he went back to Caltech as a faculty member. So I also keep on tracking his work. So I'm glad here today he agreed to give a talk on his recent work. So David has a very spectacular record. He got his bachelor degree in mathematics and physics from MIT at age of 18. Okay, this is the time probably most people just enter a college. So then he also have because he has clearly have more time. So he got a MD from UCLA on medicine in year 2013. Then he also his PhD from Caltech in applied physics in year 2011. So with Rob Phillips. For anyone who from our CPCB program, our textbook for the course Robin and Jim teach. That's it's a book, the Physical Biology of Cells by Rob Phillips and others. So Rob is David's PhD advisor. Then after that he went to Stanford to did a postdoc with Max Calvert. So there, that's where I start to know his work. So now, so his work is mainly working on a lot of things. Is try to use a lot of imaging data and try to extract information. And as I mentioned, I started to track his work, and I really want to know what his current work. So that's why I'm very glad to listen. Okay, David, it's your turn.
Thank you so much for the kind introduction, also the invitation to speak. I think in today's times with everything being virtual, I personally am very grateful for having the opportunity to showcase my lab's work. And so thank you for inviting me. And today I'll be talking about my lab's work at the interface of biological imaging and artificial intelligence. So this interface has never been brighter. The number of things that we can measure about living systems with imaging has never been greater. So signaling dynamics, gene expression, protein concentrations, all of these are now measurable with imaging. And at the same time, we've had new deep learning methods have arisen that have transformed the field of computer vision. So as these methods make their way into life sciences, they're in a very real way fundamentally changing our relationship with imaging data by letting us extract quantitative information from images with an ease that was previously impossible. And so a collection of tasks that can now be done with deep learning, which I'm using deep learning and artificial intelligence sort of synonymously in this talk. But a collection of tasks can now be done with deep learning are shown here on this slide. And this slide is really meant to represent the field's progress, not just the contributions of my group. And so these applications include cell segmentation, which is the focus of this talk, cell tracking, analyzing single molecule images, restoring noisy images, and interpreting label-free images, and some associated citations, which I find which are interesting, I think are given here. So while there's a lot of excitement about these methods, I personally think that artificial intelligence and deep learning hasn't had the same impact in biology as it's had in other areas of society. And so the reason for this is that we're currently in what I'm calling the Tico Brahe era of AI-powered cell biology. And what I mean by that is the following. So Johannes Kepler is a famed astronomer. We all learn about him during freshman physics, and his famous laws of motion, the orbits of planets are ellipses, equal areas are swept out in equal time and whatnot. And sort of these sort of general rules about how the world operates are a scientific holy grail, right? It's sort of, this sort of general understanding, these general rules, this is a large part of the reason why we all get into science. What's less appreciated are the contributions of Kepler's mentor, Tycho Brahe. And so what Tycho Brahe did during his time as a scientist is he kept meticulous notes on the identities, the locations, and the movements of the planets and stars. And these notes were compiled over decades. And it was really this data-centered effort, the collection, the curation, and the annotation that enabled this creation of general knowledge and insight. And I think it's in this phase, this Tycho Brahe phase, that AI and cell biology, that's the phase that their relationship is in now. And what that means practically is that there are some substantial data challenges ahead of us. In order for us to reap the full benefits of what artificial intelligence can do for the life sciences, we need to go through this Tycho Brahe phase. And personally, in my view, I believe that rather than avoiding these data challenges, we as a community should embrace them. If we want the spectacular things that AI can do for us, then in my view, there's really no other alternative. And the path to an AI-powered future for cell biology, really goes directly through these big data challenges. So my lab has been tackling these issues for about the last 3 years, and I think we have a sense of what the data and the annotations of the future will look like. So the data itself needs to contain information about the parts list variation. So here I'm talking about the fact that biological systems are composed of large numbers of parts. If you work in the approximation that one gene is one part, then a simple organism like E. coli will have around 3,000 parts, and mammalian systems will have on the order of tens of thousands of parts. And so the data needs to contain information about this parts list variation, and that's something that can be done using a wide variety of different genomic technologies, DNA sequencing, RNA sequencing, ATAC-seq, and whatnot. The data itself also needs to contain information about the spatial and temporal variation in living matter as well. And that means there needs to be like a imaging component, right? Imaging is our way of accessing, how do things vary in space, and then also if you're doing say live cell imaging, then how do things vary in time as well? And so having data sets that have these two different aspects to them is going to be essential to unlocking this, the secrets of how living systems operate. The annotations themselves, so these need to include dense pixel level labeling. So, getting the data for the first two invariably means that it's going to be in the form of images, and the annotations are going to require effectively labeling every pixel in every image in these large data collections. And then the second part of the annotation as well is the ability to have single cell resolution. We know that's the fundamental unit of life, and hence it would make sense that the annotations would respect that. And so a prototype of what I'm talking about is sort of shown on this video on the right. And so here we have a movie of a cell undergoing mitosis. And so the DNA is in red and the microtubules are shown in shown in green. And on the left is sort of a quote unquote annotation, which is really a schematic that Walter Fleming, esteemed cell biologist who was alive in the 1800s. This is schematic that Walter Fleming drew of this process, when he was observing under the microscope. And I think it's sort of, I'm sort of showing this because, on the right, we sort of have the spatial variation, we have the temporal variation, we have the parts list, red and green. And then on the left, we sort of have like an annotated version of what was happening in this movie. And it's some variant of this at a much larger scale is what needs to happen for the life sciences to reap the full benefit of artificial intelligence. So the data set challenges ahead are substantial, but they're not everything, and they're actually compounded by additional challenges that are associated with using deep learning methods in practice. And so these two additional challenges are models and compute, and that's actually the reason why this talk is called Software 2.0. The term Software 2.0 sort of calls attention to this reality that to use deep learning methods in practice, you need much more than just deep learning. So if I remember correctly, this term was first coined by Andre Karpathy, who's currently the head of AI at Tesla. And so we've been sort of grappling with these challenges for the last 3 years, and our experience is sort of the same as that of colleagues who are engaging in similar efforts in industry. If you want to do software 2.0 well, then you can't work on any one of these areas individually. Rather, you need to be working on them all at the same time. Effective software 2.0 requires co-development of data sets, AI models, and then also computational infrastructure for training and deployment. Focusing on one isn't enough. And each of these areas has challenges associated with them. And I just like to share what my view of these challenges are. Again, not to claim that we've solved any of them, but rather to highlight where this field is and what this community needs to focus on in the near future. So for data, challenges that exist, how much data is necessary for a given task. For a number of images, the type of annotation that you want are at the pixel level. How do we generate them in an affordable manner? Biological imaging data tends to be higher dimensional than consumer images. There can be a temporal component. You can have up to 3 spatial dimensions, and then you can also have multiple colors as well. And so how to get annotators to perform well for these challenging data sets. Data sets themselves change over time as annotation projects progress. And so then having like a governance system where you can track what changes are happening to data sets, and then which models are then derived from said data sets. Having a sensible versioning system so that you can link these data sets on the models and then the analyses that are derived from those models. I think that would be a very big step towards reproducible science. For models, understanding like which model architectures are accurate enough for automation while remaining fast enough for inference, knowing which models can actually be scaled to big data sets in an affordable manner, and then also figuring out how we can limit model size so that we can deploy entire collections of models as opposed to just focusing on individual models, optimizing that whole collection as opposed to that single model. And then for computational challenges, how can we integrate models into complete pipelines? For most tasks that folks want to be able to do, it's not entirely solved by just a deep learning model. It's invariably deep learning plus something else. And figuring out how to create and then deliver these complete pipelines. This is something I think is a major challenge. How do you migrate these pipelines to the cloud in a way that makes them scalable and affordable? And then how can you optimize these models for use on edge devices? And then lastly, there's this one area which unfortunately I won't have time to talk about today, which is how can we use software 2.0 to improve our experimental design? Now that we know what AI models can do, and once we get them performing well, how can we design better, more scalable experiments? So with that said, I'd like to share a couple stories from my lab that are the interfaces of these problems. So there's a story at the data model interface and then another at the model and compute interface. And so this data model interface focuses on how to get single cell information out of images. And it's really been spearheaded by 2 people in my lab and then 1 person in a collaborator's lab, Michelangelo. And so that's Geneva Miller, who's been a technician in my lab, Eric Moen, who's a postdoctoral fellow in my lab, and then Noah Greenwald, who's a graduate student at Stanford with Michelangelo. Okay, so getting single cell information out of images, single cell analysis is a common analysis challenge for biological imaging experiments. So our lab, I also run a wet lab, and in this wet lab, we use live cell imaging to understand how cells encode information about themselves and their environment. And so in these movies, cells express a fluorescent reporter that will report on a signaling pathway through either the amount or the location of fluorescence. And so to analyze these movies, what we need to be able to do is identify cells in every frame and then link them together over time. Once this is done, we can then convert these movies to quantitative single cell traces like this. I sort of view this as like a cell signaling version of a patch clamp. Each curve tells you like what that cell was thinking about the stimulus that it saw. In my lab, we're also interested in how these dynamic signaling patterns are converted into patterns of gene expression. And so to measure this, we perform endpoint multiplexed FISH to quantify these data, what we need to be able to do is identify the single molecule spots of these images and then assign these spots to the identified cells. And unfortunately, I won't have time to talk about our work on spot detection today, but we've been seeing some good success with the new method we've been developing that uses weakly supervised learning to solve this problem. Okay, single cell analysis is also a challenge for tissue imaging experiments. So there are new multiplexed imaging methods that make it possible to image the location and abundance of dozens of different proteins simultaneously in tissues. And so these methods involve either imaging with mass cytometry, and so a schematic of one of these methods, multiplexed ion beam imaging, is shown here, or performing iterative rounds of staining and destaining and then imaging with light. And so you either have to do one of these two things to get around the problem of spectral overlap. If you're that happens with light imaging. If you're using fluorophores, then if you're trying to get more than 3 or 4 colors in a given experiment, it's really challenging because the spectra of the fluorophores overlap. So either you will image using mass cytometry. So instead of labeling with fluorophores, you label with mass tags, and then you image through ablation, sort of destroying parts of your sample one at a time, and then taking the destroyed sample and sending off to a mass spectrometer to read out the different mass tags. Or you can do multiple rounds of imaging with light, imaging 3 or 4 different markers at a time, and then destaining and then repeating the process. And so these methods are, and the data they generate are opening new windows into the biology of both healthy and diseased tissues. And that's in part because they can measure both the spatial variation and then also the partsless variation all at the same time. And so I first got introduced to this field through a collaboration with Liat Karen, who's now at the Weizmann, and Michelangelo, who's at Stanford. And so they developed one of these platforms, multiplexed ion beam imaging. And so the data they generate is amazing. And so here, so this is here's just showing that we can visualize the difference in expression for a panel of different markers across a variety of different tissues. So here there are 4 different patients, and then different markers. P53, EGFR, CD45 identifies immune cells, double-stranded DNA identifies cell nuclei, beta-catenin, pan-keratin, and whatnot. The point isn't just to focus on any given marker; it's just to say that you can image a large number of them all for the same tissue. And so these data are very rich, but analyzing these experiments is very challenging, and single-cell analysis is a common challenge for all of these imaging platforms. And so what one invariably wants to be able to do is to go into images like this. And so here we have the images of the cell nuclei, the double-stranded antibody against double-stranded DNA. And you want to be able to say, like, okay, these pixels belong to cell number 1, these belong to cell number 2, these belong to cell number 3, and whatnot. If you know which parts of the image belong to which cell, then you can quantify all the different proteins and for each cell, and then extract information like, what are the different cell types, and what are the different spatial patterns. So as an example of this here, we just highlighted the different immune cell populations in a patient's sample. So mesenchyme and tumor are given different shades of purple, and then you have T cells, B cells, and macrophages on a color spectrum from purple to orange to yellow. Okay, and so cell segmentation is a challenge for this, is a big challenge for these analyses. And I think a big problem in the space is that all of the analyses that have been done to date, including the ones that I've been a part of, they use the cell nucleus as a proxy for the whole cell. And so this is actually inadequate for a variety of reasons. Most proteins aren't localized to the nucleus, and so if you're using the nucleus as a proxy, then you're drastically subsampling like the entirety of the cell, and then your quantification is quite inaccurate. Beyond just how much marker does the cell have, there are other analyses that one would like to be able to do. So subcellular localization is one of them, and if you're not accurately identifying the contours and the boundaries of the cells, then you can't do subcellular localization analyses. And then cell morphology is another type of analysis that is not possible using just nuclear segmentation, because again, you haven't accurately identified the contours of the whole cell. And really what's needed for this field of multiplexed imaging is a way to do whole cell segmentation in a manner that's agnostic to the different imaging platform that you're using, and then also to the different tissue type that you're using as well. Okay. So how does one go about solving these challenges? And I would say, there's a very vibrant community of people who are working on developing clever deep learning algorithms and others, both supervised and unsupervised machine learning methods for tackling these problems. And I don't want to discredit like the value of that type of work. But my belief is that the real path to creating performant models is through improving the underlying data. The reason why models don't perform is primarily because the reason why models don't perform is because the underlying data doesn't contain the information necessary to perform your desired task. For images and tissues, this issue arises as a lack of contrast in images between neighboring cells. And in live cell imaging movies, this arises because there's just a lack of temporal information in almost every publicly available training data set that's been produced to date. And so an example of how information content can improve performance is shown here on the slide. So here we have an image of cell nuclei and tissues. And so if you were to take this image and then associated training data and then train a model predict the cell boundaries using only the nuclear image, the models that you get when they do inference on images like this will produce outputs like this. And so what you'll see are all these gray areas. And so these gray areas reflect regions where the model is just uncertain about, is this a boundary or is this not? And that's because the contrast that the visual contrast that these models use to pick up on to make these decisions, that contrast is missing. What you can do if you're, say, using a multiplexed imaging method, you have additional channels that are available, and you can reserve some of those channels for structural information. So if you were to, say, take the image of a cell nucleus and then append onto it also an image of the nuclear envelope, you would then be adding back the contrast and then essentially adding back that missing information to your data set. And then if you were to, say, train and then run inference, train and then also perform inference, using a model that was trained on both that combined, here's where the here's where the cell nuclei are, and then also here's an image of the nuclear envelope, the results for such a model look like this. And so you'll see all these gray areas of confusion, vanish. And this sort of highlights the point that I was getting at. If you add back the missing information, then it's actually much easier to get models to perform. And so this is actually the same model architecture, same training conditions. The only difference is that one had access to the nuclear envelope information, the other did. Okay, and so here's where we arrive at the first story. And so working with Michelangelo, we've set out to solve this problem of whole cell segmentation in multiplexed images of tissues using deep learning. And so our starting point was to collect data from all the different platforms and tissues that are present within the community. And what we've done is we've made sure that the data that we're collecting had the requisite structural information for us to perform both nuclear and whole cell segmentation. And this means that we've compiled nuclear images and then also membrane and cytoplasm images for all the different tissues and multiplexed imaging platforms that we get access to. And so with the data, and so that was actually like a fair bit of effort, but I'd say like the labs who we contacted were actually quite willing to share their data in pursuit of this goal. And so the data annotation is actually the substantial challenge that we faced in this effort. And I just want to visually show what this challenge looks like. And so here we see a montage of images that sort of highlight these challenges. And so we'll see that cell morphology varies considerably both across and within tissues. The signal to noise and autofluorescence characteristics also vary across platforms. Cells can be tightly packed, and then also you can see that some cells will not contain nuclei, and that's because we're taking a two-dimensional cross-section through a three-dimensional object, and sometimes the cell nucleus is missed by the imaging plane. Okay, So to annotate this data, what we've developed is a human-in-the-loop approach to data annotation. And so in this method, experts, crowd workers, and AI work together to develop the data sets. The experts have the expertise to understand, how cells should be segmented, but they don't have a lot of bandwidth or a lot of time. The crowd workers have a lot of bandwidth and a lot of time, but they don't have like the requisite expertise. And so we sort of crafted this system to get the best of both worlds. Get the expertise get the knowledge of the experts, but get the bandwidth of the crowd. And so the way this works is in 3 phases. The first phase, experts create enough training data for a preliminary model, and then through that process of creating training data, they further specify the task, and they also create instructions as far as how annotation should be done. And so the initial version of data that they generate gets turned into a preliminary deep learning model that can perform segmentation. But because it's not powered by a large amount of data, it actually makes like a fairly substantial amount of errors. In the second phase, what happens is this preliminary model is used to produce predictions, and then these predictions are sent to crowdsourced workers. And the job that these crowdsourced workers are given is to correct the errors of the errors that the model makes, as opposed to creating annotations from scratch. And what we've observed is that this is actually a much better way of using crowdsourced labor. They're both more accurate and then also considerably faster at correction, as opposed to creating annotations from scratch. Once they've corrected, a batch of collection of predictions, the results are sent to an expert. The expert sort of goes over them, performs quality control, and then the final curated version is then added to the initial pile of training data. You then have a larger training data set. You train a new model. This new model is then used to create new predictions, and then the cycle repeats. And what ends up happening is that as you go through more and more cycles, the model gets more accurate, and because it's more accurate, there are fewer mistakes to be corrected, and the crowd workers end up actually performing even faster. And so this ends up scaling like quite nicely, and then at the eventually get to a point where the model accuracy is high enough where you can actually deploy it to a community for use. Okay, and so using this human-in-the-loop method, we've actually constructed a large-scale annotated training data set for multiplex tissue imaging. We're calling this data set TissueNet, which is inspired by the original ImageNet, and it's the largest collection of its kind. The data is composed of 6 different imaging platforms, and then also 6 different tissue types collected from 9 different organs. We've annotated approximately 1.3 million whole cells, and then also a similar number of cell nuclei. And I just want to highlight that the number of annotations that this data set generated is larger than the sum total that's been done by the entire community to date. There's approximately twice as many nuclear annotations than are available in all the public data sets, and then there are approximately 16 times whole cell segmentations that are publicly available as well. I'd like to highlight that this data set took approximately 2 person-years worth of work to make, but because of our approach, most of this burden was carried by a considerable number of crowdsourced annotators, and the actual total amount of time that experts had to spend annotating was quite manageable. Okay, so with this data set, we can train deep learning models to simultaneously perform nuclear and whole cell segmentation. And so the model that we've developed works by predicting the cell contours and then the cell center. And then using these 2 pieces of information, we can perform a marker-based watershed to quickly determine like which parts of the image belong to each cell. I do want to highlight that this model exists within a pipeline that normalizes images to account for variations in acquisition, and it also performs tiling and untiling so that large images can also be processed. And I raised this because I feel this is an important point, that complete solutions actually require more than just deep learning. And so we're calling this entire pipeline Mesmer, but we're in the midst of writing this work up for publication, and so there's a small chance that this name might change if our lab members continue to be imaginative. Okay. So in this case, I say, pictures are worth 1,000 words. So I just like to walk you through some of the results. And so here we have a multiplexed image of a pancreatic cancer specimen. And if we run this image through the Mesmer pipeline, the outlines are the predictions as far as like what are the contours for every cell. So this is taking the predictions for the cell masks and then producing contours from them just for the purpose of visualization. And so you can see that it actually performs really well. So we've performed extensive benchmarking of Mesmer. So as a measure of segmentation performance, we're using the F1 score. And so we're calling the F1 score is the harmonic mean of recall. If a cell is there, did I detect it? And then precision. If a cell was detected, was that detection correct? And we've compared Mesmer so to CellPose, a recently published generalist algorithm for whole cell segmentation, and also our own prior work, which we called FeatureNet, which is trained to perform nuclear segmentation in MEB images of breast cancer patients. And so using TissueNet, we split off some of TissueNet just for validation and testing purposes, and we use that data set as the basis for our benchmarking. And so we just want to highlight Mesmer was trained on the held out, on the training data set for TissueNet. FeatureNet was trained on nuclear images from a triple negative breast cancer cohort, and then CellPose was trained on the data set that was collected in the CellPose paper. And so what we see is, Mesmer is both fast and accurate. So comparing it to FeatureNet, we see that it's almost as fast as FeatureNet. So for 1 megapixel images, we can process somewhere around 7 to 8 images per second. I believe this is benchmarked on a V100. That's close to what FeatureNet can do, and it's roughly 10 times faster than CellPose. Mesmer is also markedly more accurate than any other method that exists. And so here we're showing benchmarking with these two methods. It's more accurate than FeatureNet and is substantially more accurate than CellPose. These aren't the only methods we benchmarked it against. We've also done others, MaskR CNN-based methods, Stardust, and whatnot. And then we break out the metrics by their subgroups, so recall and precision, and then also the Jacquard index, which measures the quality of the actual cell mask. We see that Mesmer is better than any competing approach when judged by all these different methods. Okay, to show visually what differences in F1 score means, we've created this visualization, and so here's a image of what goes in. So the nuclear channel and then a membrane cytoplasm channel. And then what we've done is we've taken the masks that are predicted and then colored them by the relative error in the cell area. So if the predicted cell is too large, then it's colored red. If the predicted area is too small, it ends up being colored blue. And so here we see the true segmentation, the predicted segmentation, and then here's what a visualization of the errors look like. Okay, so here's what the true ground truth segmentation would look like. Naturally, everything is white because you're comparing with the ground truth, and there's no error by definition. Here's what FeatureNet looks like, and so you'll see that there are a number of different error types. There are cells that are just too large. There are a number of different merge errors. So here in the corners, for instance. And again, this isn't surprising because it's doing nuclear segmentation followed by some expansion to approximate the whole cell. Comparing with CellPose, we see an F1 score of 0.58. We notice again that there are a number of merge errors, and we'll also notice that there are a number of cells that are missing as well. And this is CellPose's failure mode on tissue data. It misses a number of cells, and this is in large part due to just the fact that it hasn't seen a lot of tissue data because the training data set used to create CellPose doesn't have it. Okay. And here's what Mesmer produces. So we see an F1 score of 0.88. Most cells are somewhat white to a little bit blue, although we'll see like some cells that end up getting deep red because they were just missed detections. But visually speaking, Mesmer is much nicer, and these are the sort of results that make it really easy to do downstream analyses. Okay. The next few slides illustrate one of Mesmer's best features, which is its ability to generalize across tissue types and imaging platforms. So here we have a collection of images from different tissue types and disease states. And here are the predictions produced by Mesmer, along with the associated F1 score. Again, each cell is colored by how much the cell area differs from the ground truth, white being no difference, blue and red being large differences. And what we see is that the model that Mesmer is accurate across all these different tissue types and disease states. So breast cancer, colon cancer, histologically normal skin, lymph nodes, tuberculosis, ductal carcinoma in situ. Mesmer is able to segment all of these. Looking at across different imaging platforms, we sort of have the same story. And so here we have 4 different fields of view from 4 different platforms: Vectra, MIBI, cyclic immunofluorescence, and Codex. And we see that Mesmer performs equally well on all of them. And again, I just want to reiterate that this is the same model, the same pipeline, and the same hyperparameter settings used to generate all of these images. There's no need for manual fine-tuning on individual data sets. Okay, so we've compared Mesmer to human performance, and we've actually observed that Mesmer achieves human-level performance for this task of whole cell segmentation. And so we performed 2 analyses that made us conclude that. So the first is comparing with inter-annotator agreement. And so we took a portion of data from TissueNet that was held out, and then we asked 5 different expert annotators to produce annotations for them. And then what we did is we took those annotations, and then we asked, like, okay, how does the difference between any 2 annotators, how does that compare to the difference between an annotator and Mesmer? And what we observed is that this inter-annotator agreement and the annotator-Mesmer agreement, these are statistically indistinguishable from each other. So if we look at all the different tissues all the different tissue images that we asked the annotators to annotate in aggregate, we'll see that that there's no difference. And then also when we break them out by subgroup, we did not see a difference either. The second analysis that we did is that we actually asked pathologists to perform a blinded evaluation of Mesmer's outputs compared to human annotators. And so these pathologists were shown a multiplex tissue image, so the nucleus and then also the cell membrane, and then they were shown the proposed segmentations, one from a human annotator, another from Mesmer, and they were blinded to which was which. And so, and then they were asked to give a score, which of these 2 did you prefer? And so the results are shown here. And again, we'll see, so here we see like the raw data and then also a Valen plot sort of highlighting the distribution. Looking at all the data in aggregate, there's really no difference. There's no significant difference between the mean of this distribution and 0, which, leads us to conclude that there's, that the performance between Mesmer and human annotators is on par as judged by these pathologists. And even when we break them out by tissue type, the same story holds. So for some tissues like immune cells, there's a slight preference for Mesmer. For others, like colon, there's a slight preference for humans. But by and large, there's no real difference between Mesmer's output and the human annotator output as judged by pathologist preference. And so this is really exciting because it sort of means that it's feasible for us to contemplate using these sort of AI methods as more than just research instruments, but actually using them to in clinical settings and using the outputs to inform clinical decision making. Okay, so we're just now digging into like what we can actually do with Mesmer, and we're pretty optimistic about about how this is going to impact this field. And so we feel like that Mesmer is going to be pretty transformative both for tissue biology and also digital pathology. So there are a variety of different things that we can do now with Mesmer. So for instance, because we have accurate whole cell segmentations, we can accurately identify the cell type by quantifying the signal of different lineage markers and performing clustering. So this clustering, or if you're familiar with flow cytometry, the term would be gating. This clustering does have a manual component, but this manual step is much easier in the absence of segmentation errors. And so here we see, tumor marker keratin, monocyte marker CD14, and then a T cell marker CD8. So here's the raw image. here's a prediction for Mesmer, here's a prediction for the ground truth, and then if we were to quantify, okay, how many tumor cells, how many immune cells, how many monocytes, how many T cells in a given field of view, we'll see that the predictions from Mesmer and then the predictions if you were to say use the ground truth mask, cell masks, those line up really, really nicely. We can also now do protein localization analyses. So in these images, we have markers with different localization patterns. So the marker is shown in yellow, the DNA is shown in red, so cell nucleus, and then the carbon, so this is a multiplex ion beam imaging. So carbon, which sort of represents, where the tissue is shown in black. And, so here we see that Ki67 is a nuclear localization pattern, as is, phosphorus. Whereas CD44 and pankeratin, these are, membrane or cytoplasm markers as well. And so, Mesmer will produce nuclear masks and whole cell masks. We can then quantify how much signal is in each compartment, and then compare that with what would happen if we use the ground truth masks. And we see that Mesmer is very quite accurate. There's little difference with the ground truth masks. Okay, and excitingly, we're also able to now make cell morphology and observable with tissues. And the study of cell shape has a very rich history in cell biology, and we're excited about what it can tell us in tissues. And so to demonstrate Mesmer's ability to do this, we used a multiplexed imaging data set of the human decidua during pregnancy. So the decidua is a thick mucous membrane that lines the uterus and undergoes extensive remodeling during pregnancy. And this remodeling is driven by a differentiation of human endometrial stromal cells into a secretory phenotype. And so as these cells differentiate, their morphology changes from an elongated fibroblast-like shape to a globular shape. And this is due to remodeling of their actin cytoskeleton. And this morphological change does have some functional consequence. There have been some in vitro cell culture studies that have shown that the inability to undergo this morphological change is linked to severe preeclampsia, which is a condition where pregnant mothers have really high blood pressure during pregnancy. And so while this morphological change has been observed by pathologists for decades, if not centuries, it's never actually been quantified in tissues before, and that's just because we haven't had the technology to do it, but we do now. And so we collected a multiplex ion beam imaging data set of the human decidua during pregnancy, collected different time points. And to analyze the data sets, we actually created a variant of Mesmer that had a lineage-aware segmentation model. And so these multiplex imaging platforms can measure dozens of markers. And so there's a real opportunity to use lineage markers to inform segmentation models. There is a trade-off, however, because people use different markers in each experiment. And so lineage-aware models, just by definition, have limited utility because the lineage panels actually differ across different experiments. As far as like general purpose, the nuclear membrane-based model that I talked about previously has more utility. However, the lineage-aware methods have the potential of higher performance. And so, to sort of evaluate this, we annotated approximately 10,000 cells in, some of these MEB images of the human decidua, and then used them to train a lineage-aware model. And so we had 6 different lineage markers: H3, HLA-G, Vimentin, CD14, CD56, and CD3. CD3 isn't shown. And as we see in this image, which shows the results of the lineage-aware segmentation, the resulting model can, perform the segmentation like quite accurately. And again, this is whole cell segmentation, not just nuclear segmentation. Using lineage-aware Mesmer, we then processed our entire data sets, produced cell masks for every cell, and then extracted different morphology metrics from these masks. And so these metrics include the nuclear to cell ratio, the aspect ratio, different measures of radial asymmetry. And so we used the distance from the cell center and its convex hull center, and then also the difference in area between the cell and its convex hull area. And then we also looked at the perimeter squared to area ratio, and then also like the number of concavities. And these last 2 measures really try to measure spindliness. And so on the right, we have examples for each of these metrics, going from low values to medium values to high values. And then also that these these example cell masks for the different values. These are paired with like the raw data sets. And, it's sort of just meant to highlight that these metrics that we're using, they actually, they actually measure what we think that they're measuring. So we have these 6 different morphology metrics for every cell. We can then do k-means clustering on this collection of morphological features to identify morphological archetypes, and k-means sort of pulled out 4. And so cells in cluster 1 are sort of long and spindly. Cells in cluster 2 are big and globular. These likely represent the different endometrial stromal cell populations. Cells in cluster 3 are small and circular, and cells in cluster 4 sort of have intermediate values of each of these different measures. And so with the archetypes identified, we then went back to the imaging data and then grouped the images by date post-conception. So dates less than 10 weeks post-conception were considered early, 16 to 18 weeks were considered late. And then we colored cells by their archetype. And so visual inspections revealed that in the early time point, there's abundance of cells in cluster 1. And so you'll see all these red cells. These are like the cluster 1 cells. And there's visual inspection also shows a transition to an abundance of cluster 2 cells at the later time point. And so if we transition to the later time point, we'll see all of these big purple, big globular purple cells that appear. And so this change in cell populations likely represents the morphological transformation that's been observed by pathologists, and has also been observed in vitro. And this analysis really shows that we can, so we can also quantify this. We can also quantify this as far as like the ratio of cluster 2 to cluster 1 abundances in different time points. And so this analysis shows that we can harness morphological information in tissues in a quantitative way. And what's nice about morphology is that it's not biased by imaging platform. And this makes us think that there's potential to use morphology as a way to harmonize measurements across different tissue platforms. We've been hard at work building a similar resource for live cell imaging. This problem is equally challenging because there's a need to annotate entire movies rather than individual frames. And so we're currently collecting nuclear and cytoplasm movies across commonly used cell lines and annotating with single cell resolution. And I believe to date, we've annotated roughly 600,000 cell nuclei and around 100,000 whole cells during the course of this project. In addition to training segmentation models, these dynamic data sets allow us to train models that perform cell tracking and then also construct cell lineages. And so this approach was, we described in a preprint that we released last year. Roughly speaking, we treat the tracking problem as a classification problem and try to determine whether, given a frame in cell I and then a candidate cell in frame I+1, whether these are the same cells, the different cells, or is there a mother-daughter relationship because a cell division event just happened. And so our model uses information about what cells look like, what the area around them looks like, what different morphological information about cells, and uses this to perform this classification task. And we can also leverage temporal information as well because we have the movies annotated. And so the results that we've gotten with this model are quite promising. And so here we can see, and so this is deep learning to do both segmentation and cell tracking. We've benchmarked this tracking model extensively, and the real performance boost that we see is in our ability to detect cell divisions. Unfortunately, I would say that the cell tracking metrics that most people are using to benchmark these methods sort of mask the strengths and the weaknesses of the different methods that are out there. And again, I just want to reiterate, what's driving the performance of this model is the data. We're not the first people to tackle this problem using supervised machine learning. We're not the first people to tackle this problem using deep learning. But with the expansive data set that we have powering these models, we're fairly confident that even the simple model that we described in this preprint will perform either as accurately or better than most of the competing approaches that are out there. The other big issue with this, with the model that's described, is just that it's slow, and so it's actually hard to scale it to larger and larger fields of view. And we've been working on a new model that sort of uses graph neural networks to achieve similar better levels of accuracy while scaling better to large images. So this slide shows some of the challenges that we're actively working on. The first is making sure that we're actually annotating all the edge cases that happen in cell division in these live cell imaging movies. The textbook story cell division, one cell turns into two cells is not what we see in our data. Sometimes, one will turn into two, other times it'll turn into two, then back into one. Sometimes it'll turn into two, then into one, then into three. And making sure our annotation framework is flexible enough to capture all of this, all these behaviors is a really high priority. And we're also working on an integrated pipeline that can perform both whole cell segmentation and cell tracking in label-free live cell imaging movies. And so this work is underway. And if folks are sort of interested and want to contribute data, like literally all they have to do is send us an email. We're actively annotating as we speak. Okay, so while all this is exciting, I have to say that what excites me the most, more than what we've done, is sort of what this human-in-the-loop approach holds for the future. Because we've built the infrastructure to support projects like this, the marginal cost of annotating additional data is surprisingly low. If we had to do TissueNet again, the cost of annotation labor would be approximately $20,000. So that's $4,000. Well, $20,000 to $25,000, and that's the 4,000 hours required from the cloud workers. Excuse me. And then multiplied by the $5 to $7 an hour that's required to compensate people on the crowdsourcing platforms. Yeah, and so this low cost is just due to our investment in the software infrastructure to support the full life cycle of these projects. Everything from collecting data and associated metadata to curating, to annotation, to developing models and training models and deploying models. We've developed software tools for doing all of this. And unfortunately, I don't have time to go into each tool in full detail. Okay, so I think I'm close to out of time, but with the remaining time, I'd like to talk about our work at the interface of models and compute. And this really deals with the challenge of scaling analysis pipelines with large data sets, and as a consequence, making these deep learning tools available to the broad community, to the whole community, not just to the select few who have the requisite computational and mathematical ability. And so this work is done, spearheaded by our lab's first graduate student, Dylan Bannon, our lab software engineer, William Graff, and also a very talented postdoctoral fellow, Eric Mullen. Okay. All right, so models and pipelines are no good if you can't share them, and sharing them has its own challenges. Without GPU acceleration, inference times are low, but these GPUs are often in short supply. And so to share work like was shown in the previous parts of this talk, we've built cloud-native software for hosting deep learning enabled image analysis pipelines. This software can handle large volumes of imaging data as it scales cluster size to meet demand. And cluster like this that operates in this fashion can let a single user process a large amount of data, or it can serve a collection of models to an entire community. And it's really this latter use case that's inspiring us. We envision this software as a blast for cellular images. And because this cluster lives in the cloud, users can interact with it by submitting their images for analysis and receiving the results. And so it effectively changes the relationship that life scientists have with imaging data. It can effectively be treated like sequencing data, because it operates in a similar fashion like BLAST does. And so we built plugins. Okay, so we have this cluster. It's being hosted by DeepCell publicly on deepcell.org. And we built plugins for different software packages, so like ImageJ and QPath, so users can do exactly what I just said. So you can open an image in ImageJ, and using the plugin, you can send the image to this cloud server. It'll run the specified analysis pipeline, and then you will get the results. You'll get the results back, and can be visualized all within ImageJ. And so this deployment model solves many of the pain points of using deep learning. The only downside is that this solution comes at the price of network latency. You have to submit your data, and then you have to wait for the data to come back through the network. So this work was recently published in Nature Methods. We demonstrated that the software can scale to handle millions of images while also constraining costs, and data sets of approximately 1 million images can be processed in several hours for about $200. And so I personally think that this is quite affordable. You can actually get the cost down by a factor of 2 based on your cluster configuration. And I think the analysis in this paper, sort of puts to rest any concerns about the affordability of cloud computing for this use case. We've built the software to be flexible and user-friendly, so we can construct pipelines that use multiple models. And so, here we've built a pipe. Here the schematic shows a pipeline that performs segmentation and then also tracking, and then returns the results. And these pipelines, again, are made accessible either through a web portal, plugins, or through a command line interface. An example of this is shown in here. And so this is, we recently hosted a tutorial at an HGMI workshop, and a colleague at an imaging center in La Jolla sort of loaded his image of mouse tonsils in QPath, submitted them to Mesmer, which is hosted on deepcell.org, and then received the results back all in QPath. And I would like to say that all of the software that I've talked about here and software that I haven't talked about here is available for free through open source software libraries. It's free completely free for nonprofit use. And so there's software for annotating biological images. So we have a software package written called DeepCell Label. There's the data sets themselves. The Mesmer data set isn't available, but the early version of the live cell imaging data set is. And so we have a data set module, DeepCell data sets, that hosts annotated training data that we've created. We have a software library, DeepCell TF, which hosts implementations of deep cell models for doing single cell analysis, both the segmentation and cell tracking. And then for deployment and inference, the deep cell kiosk will let you spin up one of these scalable cloud clusters that are tied to your own resources. And deep cell applications has pre-trained models for doing single cell analysis, including Mesmer, and then also the cell tracking model that was shown here. Okay, and so all of this is available at my lab's GitHub under permissive, modified Apache licenses. All right, and so that, and so lab's GitHub here, github.com/Valen. If you want to just try some of these models on your own data, we have the models are being hosted at deepcell.org, and we have tutorials that sort of walk folks through the whole ecosystem at this Intro to Deep Cell repository here. And I would just harken back to the original slide. We sort of have these 3 different pillars of data, of models, and then compute. And what we're trying to do is create solutions for the broader biological imaging community at each of these pillars, from the data side, the model side, and then the compute side as well. So that concludes my talk. I would like to, I know I'm almost over, but I'd like to thank the folks who actually did the work. The data annotation work, again, has been spearheaded by a postdoc in my lab, Eric Moen, and a fearless technician, Geneva Miller, along with Noah Greenwald in Michelangelo's lab. This is a collaborative effort, and an equal amount of credit goes to Mike and his folks, and Liat Karen, who's a new PI at the Weizmann. She also deserves a large amount of credit for this project's success as well. Need to give a big thanks to our lab software engineers, Will and Tom, who work day and night to make sure that our software is robust and performant. Emily Laubscher and Maureen Schwartz are graduate students who have contributed to the deep learning methods development. And then also Dylan Bannon, who is responsible for a large part of our cloud computing work. And lastly, I'd like to thank our sponsors, most importantly, the Kersey Foundation, Rita Allen Foundation, Susan E. Rowley Foundation for supporting this work. And then also NIH and Paul Allen Foundation through subawards, and then various centers at Caltech for their support. And with that, I will stop talking and open the floor for questions.
Hi, David, thanks for a nice talk. So can you see the chat? There are a few questions.
Yeah.
Can you, yeah.
Yeah, okay. So let me, let me keep the slides open for now. Okay. Cheaper than a PhD student. Yes, I would say it's not just cheaper, but you actually get the results back faster because you can have multiple people working on the annotation at the same time as opposed to just 1 or 2. Why isn't TissueNet data available? That removes a whole pillar. That's right, and I would love to make it publicly available. The reality is we haven't submitted any of this work for publication yet, and we haven't even posted a preprint. And so it's be premature to release the data set until that happens. I would say there is some concern on our part to make sure that we sort of get fair credit for like the large amount of work that we've done. And what will likely happen is, after we submit this for publication, we get the first rounds of reviews back. If it's clear that, it's going to go in, then we'll probably release the data set by then. I would argue quite strongly that sort of asking us to, release everything before we've gotten, our due amount of credit, that would be unfair. I would say like this work was actually done, without any support from like the large funding institutions, like the NIH, and it's not like we haven't asked the NIH for money. So after we sort of get the credit in the form of publication, I'd say like we're definitely going to release the tissue net for use, and I think it's really going to spur a lot of methods development, and I think it's also going to help people answer some, I'd say some fairly open questions, as far as how large of a data set do you need, to get good performance? I'd say like it's gonna answer that. Will Mesmer be open source? Yes. The model itself is actually like available now. We have a pre-trained version on our lab's GitHub. We also have, we also have like Mesmer hosted on deepcell.org. Thank you for the nice comment about the work. Impact of cell segmentation errors have on cell phenotyping. It has a large impact. I would say, the phenotyping is very hard if you do not have accurate cell segmentations. Having accurate cell segmentations isn't enough though, and that's actually like an open problem. How do you properly phenotype and identify cell types once you have accurate segmentations? That's what we're working on. How do we get the ground truth? Again, it's this human in the loop process. Experts create the first version, create a preliminary model, and then crowdsourced workers correct the errors that are present when you run the model to perform predictions. And then the corrected data sets get added to the original pile, and then you sort of repeat the process. And so the final data ends up being like a hybrid, human expert annotated, crowdsourced annotated, and then also like model predictions. I sort of view the data sets kind of like a cyborg, because part generated by AI, part generated by humans.
Okay, Amir, you raised a hand. Can you just ask the question?
Yeah. Thanks for the great talk. So I think this is a great resource. One question I had is, do you plan or plan on working on providing a benchmark kind of data set that might be something like a competition, just like there's ImageNet, and to basically drive community optimization of standard benchmarks? Because like one of the main issues in these fields is when we have data sets that are always publicly available, publications and work from the community might be incremental and not actually.
Might what? Sorry, you started breaking up.
Can you hear me now?
Yeah, I can hear you.
Yeah. So like Netflix is a famous example for movie recommendations where because they had a held out data set, it was better than another data set like the Movie Lens, where they didn't have it held out, and they showed that basically taking very baseline benchmarks from Netflix Prize, applying it to Movie Lens could actually beat the last 5 years of state-of-the-art results on Movie Lens. So I think like this is a great opportunity to do something similar here. Do you plan on doing that and providing such like a
so the data set itself, like we're planning on releasing, and so folks will be able to use it to like benchmark as like benchmarking, and I do think like that's something that's not fully appreciated. If you do not have annotated ground truth, it's really hard to know like what's progress and what isn't. And so I agree, and that is one reason like why we want to release it. I would say like as far as releasing, as far as like managing a competition, like we have no plans to do something like that. And I think it's really one, it would be like a fair amount of work. Two, I would say like for this challenge, where the performance of models are already at like human levels performance, and so like how much additional performance would you need? And then three, I would say, what's the purpose of like competitions? And I'd say like there's a difference in community as far as like how the machine learning community approaches competitions and how the life science community approaches competitions. The machine learning community will actually invest in the competitions and make sure like they're run regularly every year to sort of keep track of progress and to hold folks both accountable and honest about like how well their methods are doing. What has happened as far as competitions in the life science community is that that doesn't happen. The competitions are sort of end up being one-offs, and then the labs that run them sort of take the results and go off and write like a Nature Methods paper about them. And so the credit that one would expect as far as like what advance gets made, that actually doesn't go to the the methods developers, the folks who take part of the competition. It gets taken by the folks who host the competition. And something about that is someone who likes programming and writes these methods, something about that does rub me the wrong way a little bit. If we were to do a competition, it would definitely be much more in the line of what the machine learning community has done, just like regular competitions. But I'd say like for this challenge, I'm almost of the opinion, like what's the, I feel like, how much better than human do you need? Yeah.
Okay, I think it was quite late, probably. Yeah, if people, if you still have questions, I think you can just email to David, right?
Absolutely. I love answering questions.
Okay, good. Okay, let's thank David again for this very nice talk.
Thank you.
Okay.
All right. I think I have meetings after this, so I will see folks in the next Zoom room.
Yeah, we'll see you soon.